So let's take a look now at the other means of transportation across the membrane, the more active forms, either through facilitated movements or through active transporters, endocytosis, and then finally we'll look at the means of exocytosis. So the first one we have to look at is the facilitated movement of materials. Movement of materials this way is going to go through gated pores. When we look at gated pores, the first type of gated pore we have is what's referred to as the voltage gated channel. These are going to be open and closed based off of changes of the membrane potential. That is the relationship of positive and negative ions across the membrane that will either open or close the pore to allow for movement of materials through the channel itself or through the pore itself. The second type of gated pore is the ligand gated pore. The ligand gated pore is going to undergo what's referred to as a conformational change when a messenger protein or a metabolite, a hormone, a neurotransmitter, or some sort of uh, chemical sits into the protein itself, and that causes the protein to do a change in its shape. The change in its shape will then go about opening the channel to allow for the materials to move across. The last type is what's referred to as the mechanical gated. In this case here, the membrane itself gets stressed, and when the membrane itself is stressed, the pore is opened up due to that mechanical stress. So let's take a look at these transporters. We'll look at it in terms of the symport and antiport movement through the facilitated transported motions. So the first of these is going to be the symport. What we're doing is we're moving it in the same direction. And so if we watch this, we have a sodium and a glucose symportation. So as sodium opens up the channel as a ligand, it will allow for the glucose to follow along into the internal side or the intracellular side of the membrane, all due to the conformational change of the transporter simply due to the interaction with the sodium ion. This is the transporter that we see utilized in terms of rehydration, utilizing the sports drinks, the electrolyte and carbohydrate sports drinks that are used for rehydration purposes. Is the antiporter. And so in the antiporter, what we have is we're going to have materials move in opposite directions across the pore once the pore is opened. In this case here, we're looking at a sodium hydrogen antiporter. This is something that is very similar to what we see when we look at acid pumps within the secretory cells of the stomach, where we're going to be moving similarly charged ions across the membrane based off of the conformational change that we see due to the initial interaction. So those are the facilitated transporters. The other type is active transportation. In active transportation, we're actually going to be utilizing the ATP molecule to go ahead and phosphorylate or kinase the transport protein in order to move ions, in particular sodium and potassium, which is the most common of these active transporters. So what we do is we see 3 sodium ions lock onto the transporter. What this does is this opens up a binding site for the ATP. When an ATP binds on, it strips one of the phosphates, which energizes the protein, causing the protein to do its conformational change. What this does is opens up an electrical attraction for potassium. And when potassium locks on, what it does is it changes the electrostatic nature of the protein, which causes the phosphate to dislodge. When the phosphate dislodges, it causes the protein to reconfigure to the resting state. And the cycle will continue as long as sodium and potassium need to be pumped across the membrane, which it constantly has to, as the cell has enough ATP available to allow it to proceed. So that works for things that are small. But what about things that are large, or things that need to be trapped and then brought in, or things that need to be released? This is where we get the 2 processes known as endocytosis and exocytosis. So let's go ahead and let's watch an animation here as to what endocytosis and exocytosis happens to be.
The substances taken in by single-celled organisms are often particles or large polar molecules that cannot cross the hydrophobic plasma membrane. Many single-celled eukaryotes employ endocytosis to ingest such food particles. In this process, the plasma membrane extends outward and surrounds the food particle. Cells use three major types of endocytosis: phagocytosis, pinocytosis, and receptor-mediated endocytosis. What the cell takes in is liquid. It is called pinocytosis. Specific molecules, such as low-density lipoproteins, LDL, are often transported into eukaryotic cells through receptor-mediated endocytosis. Molecules to be transported first bind to specific receptors on the plasma membrane. The interior portion of the receptor protein is embedded in the membrane. The protein clathrin coats the inside of the membrane in the area of the pit. When an appropriate collection of molecules gathers in the coated pit, the pit deepens and seals off to form a coated vesicle which carries the molecules into the cell. Exocytosis is mitosis. This process results in the discharge of material from vesicles at the cell surface to the outside of the cell.